As a bit of background so that we're all kind of on the same page, the microbiome is the collection of microorganisms that can include bacteria, protists, fungi, but oftentimes we're talking mostly about bacteria that contributes to the overall health of the host. And of course, by contributing to the overall health of the host, digestive health, immune health, skin health, in birds, feather health, lots of implications for the microbiome in affecting its host organism. Because the microbiome can have such an effect on the host, particularly various host phenotypes, it follows that the microbiome could play a role in host evolution as well. The microbiome can be acquired in a number of different ways, vertically from an organism's parents, from the environment, things they touch, etc., and of course through social contact. In our previous study, we were interested in looking at the Uropygial gland of the dark-eyed Junco. And the reason for that is because previous work had shown that microbes found in the Uropygial gland can produce volatile compounds. And those volatile compounds could then be detectable by other birds as a type of chemical signaling. What we found in terms of the microbiome of the Uropygial gland was that the alpha diversity was lower in females, though that wasn't significant. So basically, females had fewer taxa of microbes found in their Uropygial gland, and in the migratory subspecies, and these data were significant, when compared to the resident subspecies of dark-eyed Junco. The interesting thing that we found as well was that alpha diversity, the number of taxa, decreases over time in captivity. So that has implications for how captive studies might need to account for the microbiome, because we did see that significant decrease in diversity over time. We also looked at community composition, and we found that the community composition of the Uropygial gland microbiome was different between migratory and resident birds, and that community composition also changed between the various time points of the experiment. And we found that overall, the community composition, the communities got closer together. So the Bray-Curtis dissimilarity, it decreased over time in the experiment because these organisms were captive, because they were housed with one another. We saw that their microbiomes started to become more similar. Again, implications for how captive studies should be designed to account for the microbiome. In terms of how the Junco Preen microbiome affected the volatile oil composition, we saw that the microbiome composition was weakly correlated with overall Preen oil abundance. So the significance of this work is really the idea that characterizing the microbiome of animals is important for trying to understand how the microbiome can affect phenotypes and potentially change fitness. But specifically with this study, we're learning how captivity can affect the microbiome, and how scientists will have to, going forward, really think about how they design their experiments, because these alterations in the microbiome and the potential downstream effects on the overall health of the host organism can be really important when considering what you're trying to measure experimentally. The goal of the particular study that I'm presenting at AOS is to identify how sex, subspecies, and captivity can lead to changes in the cloacal microbiome of these dark-eyed Juncos. So the experimental design, briefly, birds were captured, and at capture, they were sampled. Both the preen and the cloacal areas were sampled with a swab. And for the first 4 days, they were housed in same-sex, same subspecies flocks. So migratory males, migratory females, resident males, and resident females. They were sampled after those 4 days and moved to mixed sex pairs, either with the same subspecies or with different subspecies. So you can see that design here. They were sampled again after 4 days in the pairs and after 8 days in the pairs. After 8 days in the pairs, they were moved back again to same sex, same subspecies flocks, where they were sampled after 4 days and after a total of 8 days back in their same sex, same subspecies flocks, after which they were released. When we were looking at the overall community diversity, right, that alpha diversity, we didn't really see any significant differences in the cloacal microbiome between migratory and resident or between male and female birds. But what we did see, just like with the preen microbiome, was we saw a decrease in the diversity of the microbiome over time. So we have observed ASVs. This is basically like the observed sequence variants, the number of taxa, Shannon diversity, and the Simpson's index. And again, they all show a decrease that is significant, with the exception of the Simpson's index is not a significant decrease. What I found really interesting about looking at these alpha diversity metrics was the decrease in overall diversity after the first 4 days in captivity. That slight increase when the birds were moved from same sex, same subspecies flocks into their mixed sex, same or different subspecies pairs. And then again, just kind of that steady decrease after that time in captivity. In terms of community composition, we used Bray-Curtis dissimilarity to determine how similar or dissimilar the microbial communities were. And what we found is that at time 0, right, at time of first capture, really no differences between the sexes, between the populations, or between what we were calling the flocks. However, we do see some significant differences between the various time points. And just like with our alpha diversity, we see an overall decrease in Bray-Curtis dissimilarity over time. And basically what this means is that over time in captivity, from the time of capture to the time of release, the microbial communities of the cloaca are becoming more similar to one another. And just like with the alpha diversity, we see that significant decrease in the dissimilarity between time of capture and that first round of captivity. So to show you again what the experimental design is down here, we see kind of an increase, right? An increase in the diversity of the community, or a dissimilarity of the community when they're moved into their pairs. We see again a peak in the dissimilarity, so the communities become a little bit more different when the birds are moved from their pairs to their same sex, same subspecies flocks. And after they've been in those flocks for some period of time, we again see that decrease in community differences. So overall, the communities become more and more similar. Again, just highlighting how important it is to consider the microbiome, the potential effects it could have on the health of the host and your experimental design when you're doing captive experiments. Finally, we did compare the preen and cloacal communities, and the trend, of course, at time 0, the trend is that the cloacal microbiome is more diverse than the preen microbiome. The data were only significant when we're looking at the observed taxa. They were not significant when looking at Shannon or Simpson's index. And overall, the community composition between the cloacal and preen microbiome is significantly different. And here are our funding sources. So if you have any questions, feel free to reach out to me or any of the other members of this study.